So, good afternoon, everybody. I will start my class today, and today I will be talking about genetic code. This is the chapter which you have known. You know that the genetic code is read as triplets. They're read as triplets. The genetic code is read as triplets, and resultant the genetic code codes for there is a specific amino acid for a genetic code, and it is present on the mRNA molecule. With this, I will share my screen. Okay, so we are talking about देखिए के एलो nobody. Okay, fine. So we are talking about genetic code. So the sequences of nitrogenous bases of a given segment of DNA actually has been found to be identical to the linear sequence of amino acid. The collinearity of protein molecules and DNA polynucleotides, it has given the clue that the specific arrangement of 4 nitrogenous bases A, T, G and C in the DNA polynucleotide chain somehow determines the sequence of amino acid in protein molecules. এর মানে দাঁড়াচ্ছে যে যে সিকোয়েন্স অফ যে ডিএনএ বেসিসগুলো ছিল এ টি জি সি ডিএনএতে সেটা এমআরএনএর সঙ্গে কোরিলেট করছে, আর এই সিকোয়েন্সের ওপরে একটা পার্টিকুলার সিকোয়েন্স অফ ডিএনএ হলে একটা পার্টিকুলার অ্যামাইনো অ্যাসিড সিকোয়েন্স আসছে, এটা দেখা গেছে। তার মানে একটা কোরিলেশন ছিল। So how do you then define the genetic code? The genetic code is then defined as it is a set of rules by which information encoded within the genetic material bolche either DNA or mRNA is translated into protein. So the information which is present in the DNA which gets converted into the mRNA, that somehow is converted into the proteins. So this is because, and it is a set of rules. So it is a set of rules by which this information from DNA is converted into proteins in living cells. And এর জন্যে you need a machine known as the ribosome. You have known about translation, and this ribosome, it links amino acids in an order specified by mRNA using tRNA molecules to carry amino acid to read the mRNA 3 nucleotides at a time. তার মানে there are 3খানা নিউক্লিওটাইড পরপর পড়ে, সো একটা এমআরএনএ সিকোয়েন্সে 3 করে there are lot of mRNAs প্রেজেন্ট থাকে। তারপরে কি করে এরা এই রাইবোজোম আর টিআরএনএ ওরা কাজ করে, টিআরএনএ এর পেছনে একটা পার্টিকুলার কোডের জন্য একটা পার্টিকুলার অ্যামাইনো অ্যাসিড আসে, তাই পরপর তারা জুড়ে যায়। দিস ইজ হাউ ইট টেক্স প্লেস। So basically it is very simple, and there are 64 entries. So roughly দেখা গেছে there are 64 codon. এবারে why is it a genetic code a triplet? So there are 4 letters G A C U mRNA, T থাকে না তাই জন্য T এর জায়গায় U. So there are 4 letters G A C U. These 4 letters can be combined to form 64 genetic words consisting of 3 letters. So like I told, there are 20 amino acids and there are 4 nucleotides A, U, G, C, and now so this must code for this. So if one A codes for one amino acid, say for example, then 4 to the power 1, that is equal to 4, And you can only so with 4 letters of A, U, G, C, you can code only for 4 amino acid, but there should be 20. So genetic code is not a singlet. It can be, if it is 2, then it is 4 to the power 2. Say AT or GC or CA. So how many combinations can you get? You can get with this 4 square, that is 16 combinations. Still this is not enough. The number of amino acid is 20. So this is out of question. Then the next thing comes, if it is read as 3, what happens? 4 to the power 3. That gives a combination of 64. This is good enough. 64 combinations, 20 amino acid. This can cover. So this has been selected. 4 to the power 4, that is become something 264. That is too much. Why will there be so many? Five of them. Why will 5 pentalettes? Why will it be required? It is not proper quadrilettes. If it quadrilettes or pentalettes, it is not ready. It becomes, the combination is too much. You have to code for only 20. So the minimum which is required is 3. And this was later proved in an experiment by Nirenberg and Khorana, that the genetic code is read as triplets. So this is what I've explained over here. So it is read as triplets. So by Marshall Nirenberg and Hargobind Khorana, along with Robert Hawley, to determine the nucleotide sequence of several tRNA. Now, what are the characteristics of a genetic code? The genetic code has got a triplet nature. That is, it is read as 3 in 3. So, 2 amino acids, methionine and tryptophan, they have 1 codon each. Okay, 9 amino acid, ASN, ASP, cis, GLN, GLU, his, lys, phenotype, they have 2 codons each. There are 2 codons which can code. There are 2 different codons which can code for ASN. There are 2 codons which can code for ASP. There are 2 codons which can code for CYS. There are 2 codons which can code for GLN. There are 2 which for GLU. And also the same for all these things. So total for first 1 codon each. So 2 has been taken. Now 18 has been taken. 2 into 9 amino acids present. Yeah, isoleucine has got 1. So 3, and then amino acid, this isoleucine, 1 amino acid, it has got 3 codons. So 3 of the codons has been taken over here. And then the amino acid, all these amino acids, they have 4 codons. So since there are 5 amino acids, 20 they have been taken. You have the amino acid, these 3 amino acids, they have 6 codons each. So 6 3s are 18 of them taken. And there are 3 termination codons. These codons will stop. So it is 3. So all with this 3 codons, you have so many combinations. 3 terminator codons. So 3 so if you add all of them, you see that they can get, you get the 64. So by calculation also you can get that genetic code is read as triplets. You can get 64 combinations which you have seen. And there are 64 combinations which are eventually used. The next thing is the pattern of the genetic code. Amino acid with similar structural properties tend to have related codons. For example, aspartic acid has got GAU, GAC, and glutamic acid, which is similarly acidic, they have got GAA and GAG. So basically the last one is different, otherwise it is same. So this can have, there can be problems because of this later. Because you will know that the third base is a wobblet, but in these cases it is not wobblet, wobble. Similarly, the codons for amino acid phenylalanine, UUU, UUC, tyrosine, UAU, UAC, and tryptophan, UGC, they all begin with U. So you can find that there are some patterns in similar amino acids have similar genetic codes. Another thing is degeneracy. Degeneracy means, for example, all these amino acids, each of them has got two codons. So that means they're degenerate. That means any one of them, one of these two will code for the same amino acid. So you have two codons for ASN and any one of them will bring ASN. So that's how it goes. Therefore, it is genetic code is degenerate. Non-overlapping. That means it is not like this 1, 2, 3 again doesn't overlap like this. It doesn't happen like this. Neither it happens like this. And two of them are like this. Neither the genetic code is overlapping. It is exactly once one finishes, the next one starts. It is non-overlapping. Then you have the codon pairing. So leucine, if it is GAG, and in the mRNA, if it is CUC and CUU, the last base is wobble. It is not a very firm pairing. লদভদ করে, wobbling মানে হচ্ছে লদভদ করে. It is not very firm. So the third in this case, both of this case leucine will come. Even if it is GAG or even if it is CUC or even if it is CUU on the mRNA, this is codon anticodon pairing. This is the anticodon here and this is the codon here on the mRNA. So whatever be it, leucine will come with either of these two because the third base is weak. And universality. In all organisms, it is universal. It is the same. There are certain exceptions. Those exceptions in mitochondria and some bacteria, you have to read about those exceptions. Then polarity. Once it starts like this, always it is going to be like this. Only the coding strand will be used. So there is always a polarity. The genetic code has polarity. That is, the code is always read in a fixed direction. That is in the 5' to 3' direction on the mRNA. It is not read in the other way, other direction, obviously, because the ribosome moves from the 5' to the 3' direction, and therefore it is read from the 5' to 3' direction as triplets. So it has got a polarity. And chain initiation is methionine, and that is always AUG. And it will bring in methionine. There are some exceptions, but it is like this. And terminators, there are three: amber, opal, ochre. So there are these UAA, UAG and UGA. These three, they don't code for amino acid. But when these three codons come, immediately the ribosome gets the message that I must leave the mRNA and the translation stops. So therefore they are known as stop codons. So at the end of each protein, there is a stop protein mRNA, there is a stop codon. That stop codon is not translated, but before the stop codon, only till then the amino acids are added. Then of course I explained what the wobble hypothesis is, that the third base is not specific, and this is the genetic code. You have to remember this genetic code. First letter, second letter, third letter. Take one from here, 1, 1, 1. So this becomes UUU. Then 1, 1, then UC here. So UUC. So like this, which genetic code codes for which genetic amino acid, you can find it from here. I will upload this today in the Google Classroom. So this ends my class today on genetic code.
Thank you, sir.
People have got any questions?
না স্যার, দেখবো ভিডিওটা তারপরে।
আচ্ছা, ঠিক আছে। রেকর্ডিংটা আমি তাহলে পাঠিয়ে দিচ্ছি। তাহলে শেষ করছি, আবার 12:30 আমি কলেজের ক্লাস চালু করব।
ঠিক আছে স্যার, ঠিক আছে।
ঠিক আছে, ওকে।